Assalamualaikum and hi, my name is Nur Azmina binti Muhammad Amin, and in this video, we will go through an experiment on the rate of photosynthesis. The objective of this experiment is to determine the effect of the light distance on the rate of photosynthesis. Photosynthesis is the process by which plants use sunlight, water, and carbon dioxide to create oxygen and energy in the form of sugar. In this experiment, we need to see how the distance of light source could affect the rate of photosynthesis. With the global warming increasing each year, it is important that we find a way to channel the excess energy or sunlight that we are getting to a place that will produce something that benefits us. The hypothesis of this experiment is the closer the distance of the light source, the higher the light intensity, the higher the rate of photosynthesis. The materials that will be used in this video are your chosen type of plant or leaf, which I use hyperangea leaf, a syringe, sodium bicarbonate, distilled water, beaker, hole punch, torchlight, ruler, and timer. Now, let's go through step by step on how to undergo this experiment. First, I've already chosen a species of plant. And next, I need to punch the leaf to get 10 leaf discs from the plant. Next, remove the plunger and put the leaf disc inside the syringe and make sure it is all accumulated at the bottom. Next, insert back the plunger and you must make sure to be careful to not damage the leaf. Moving on, mix 0.5 grams sodium bicarbonate with 50 ml of water and draw about 6 to 8 ml of bicarbonate solution into syringe. Leaf disc should float in the solution. Next, we're going to expel the air by holding the syringe with the tip up and gently pushing on the plunger. Next, plug the tip with your finger and gently pull the plunger to create a slight vacuum. There should be tiny bubbles produced and some of the discs should sink. We're going to repeat this until all the leaves sink. Next, we are going to pour the leaf disc into the bicarbonate solution beaker and set up the light 3 cm above the solution, turn the light on and set the timer. Record the number of floating leaf discs until all these are floating. Remove the light and wrap beaker with aluminium foil or put on dark space. Repeat steps with same leaf but 10 cm distance of light from the beaker. Based on the results obtained, the light distance of 10 cm take longer time for the leaf disc to float compared to the light distance of 3 cm. First question, how does the suction help the leaf disc to sink? Leaves have air spaces between the cells. When gentle vacuum is applied, the air is forced out of the leaves and replaced with the solution. Hence, the leaf disc will sink. The second question is, if you conduct the experiment without baking soda, what will happen to the leaf disc and why? Baking soda serves as a source of carbon dioxide for photosynthesis. Without baking soda, the leaf disc would not rise to the surface as there is not carbon dioxide present. And the absence of carbon dioxide will not allow photosynthesis to happen, and therefore no oxygen accumulated in the air of the leaf disc. Third question, why do the leaf discs in the baking soda solution begin to float? In the presence of light, which is table lamp, and carbon dioxide in baking soda, photosynthesis occurs and leads to accumulation of oxygen in the spongy mesophyll. This will increase the buoyancy of leaf discs and causing them to float. And fourth question is, what happened to the leaf disc in the dark? Justify your answer. The leaf disc will sink as the absence of the light stops photosynthesis and no oxygen will be produced anymore. Accumulated oxygen will be used for respiration and the buoyancy of leaf disc will decrease and eventually depleted. Hence, the leaf disc will sink. Conclusion. Photosynthesis occurs in the presence of light, water, and carbon dioxide to release glucose and oxygen. Absence of any of the reactant will result in no photosynthesis happening. In this experiment, the further the light moves from the plant, the fewer oxygen bubbles are produced by photosynthesis, and it will take longer time for leaf discs to float.